Hello? Hello? Okay, perfect, it works. Welcome, ladies and gentlemen. As the title suggests, we will be doing a science information stream. This one is going to be about cellular biology, as the title says, about the neurons and glions, and without further ado, let's get straight into it because we have a lot of content to cover. So, we first begin with a preamble, and the preamble is basically to know and to better understand what we have to work with. So, what do we know about neurons as a whole? Well, think of neurons as being the attack damage carries of the human bodies. They're the ADCs, right? And alongside them, who says ADC also says supports, and that's where your glial cells come in. So neurons are the conductors of the human body with many glial cells supporting them. Starting with the neurons, the average neuron composition in the brain is about averaging 100 billion of them. So that's a lot of them. Although, and here's the interesting fact, there are, like I said, around 100 billion neurons, but they only compose only 10% of the brain. That's crazy, right? And unfortunately, once neurons reach maturity, they just do not divide anymore. So if you lose neurons, they just won't grow back, right? Because they won't divide. You know, they can't regenerate anything, right? And then we move on to the neuroglial, or the neuroglia, sorry about that. The neuroglia are actually, like I said, the supporting cells. They take care of the neurons. And of course, since neurons compose 10% of the brain, well, the rest is composed of the neuroglia, which obviously is 90%. And also, in this case, since neurons do not divide, neuroglia do divide in order to support more of the cells, of the neurons, essentially. So that being said, we can finally move on. First of all, we're going to start talking about the neuroglia. We have about 4 of them to cover, 4 types of them to cover, technically 6, but 2 of them, which are the astrocytes and amphisites, and number 4. So yeah, number 2 and number 4 practically do the same thing, even though they're like 2 different types of cells, which are very distinct. But they do it the exact same thing, so we kind of regroup them as the same type. So like I said and stated before, neuroglia are the support cells of the neurons, which are comprised of Ependymo Sites, astrocytes and amphisites, which are the support cells, if I'm not mistaken. I don't really remember what they're actually called. I think they're satellite cells. There we go. That was the name I was looking for. Otherwise known as satellite cells. Microgliocytes, which are the immune cells of the nervous system, and then finally oligodendrocytes and neurolemocytes, otherwise known as Schwann cells, for those if that name is more common for those who know it by that name. So without further ado, let's cover them all before getting to the neuron, because they, like I said, they are the support cells and they're the ones that do the most important job, which is helping our attack damage carries, which are the neurons, to do their job, basically. So, we start first with the Ependymo Sites. The Ependymo Sites, which are like they're basically the membrane. The membrane tissue that separate, they're like a barrier, right? They're like they're like a border, right? Between the inside of the nervous system and the outside of the nervous system, if I could put it that way. Basically, ependymocytes make up the ventricular lining of the central nervous system, right? Of the CNS. And yeah, basically the CNS. The Ependymo Sites play an important role in the projection of cerebral spinal fluid. And like I said, not only do they make the border of the central nervous system, but they will also create that cerebral spinal fluid via, like, via the blood vessel. They're going to take the blood, some of the blood, mix it up and metabolize it inside of their own cell and then spew it out as cerebral spinal fluid, if I can put it in a nutshell like that. That's what basically they do. And then finally, they also help in the transport of substance between the cerebral spinal fluid and the brain parenchyma. So basically, as you see in the, that's a drawing of an Ependymo site. But you also have the sting section of how it actually looks under a microscope, which is right below. And the following glial cells will also have the same function. You'll be able to see what they look like under the microscope or under a photon microscope. And you're going to have like the simplified drawing at the top. Moving on to the astrocytes and amphisites. What you're seeing here is a drawing of an astrocyte, and also the same thing for a photon image of an astrocyte as well. Satellite cells, otherwise known as amphicytes, or the other way around, however you want to put it. I couldn't find any images of them. But again, like I said, they're grouped together because they basically do the same thing. With astrocytes being the most, having the most amount of numerous, there's a very high amount of them, basically, in the central nervous systems, while mostly amphicytes are found in the peripheral nervous system, right? Basically, astrocytes provide axon guidance and synaptic support. They control the blood-brain barrier and blood flow towards the neurons, basically, because one thing about the blood, basically, the plasma, is very toxic if it ever gets inside of the brain or the neurons. If it actually touches the neurons, it's completely toxic. So you got these astrocytes really regulating what goes in and what goes out of the blood-brain barrier. And that's what they make up for it, right? Basically, they're like the TSA of the brain, right? They're like the security of the TSA, right? When you go to the airport, you have to pass TSA, right? That's what they basically do. And then finally, astrocytes and amphicytes both regulate the metabolism and detoxification of neurons, basically. Moving on to the microgliosides. Like I said, we're really going to go really quickly. This is like a very basic introduction to the glial cells. We're not going to go like in depth of what they do, because like I said, I'd rather get to the neuron part. The neurons are the most important part of this video. The glial cells are, like I said, they're like the support cells, and obviously supports never get no love, so we got to move on to the attack damage carries. Microglios, even though they're support, even though they're like the immune cells, when, as in, they're like the defenders of the central nervous system, they basically act as a support as well, because you really want to defend your neurons. Your neurons are like the type of organisms that dictate what happens in the human body, basically. With that being said, the microglia, they're considered to be a type of macrophages that act as the main defense in the central nervous system. Like I said, they're very close to what a macrophage does. And with that being said, an interesting fact out of that, there are also having the ability of a macrophage, meaning that they are able to phagocytose neurons in case of neural damage. So for, let's say, necros, for example, necrosis, a neuron necrosis, which is a horrible thing that happens, it's like the destruction of a neuron. Basically, if the neuron spews its like insides, its cytoplasmic inside outside of itself, that's pretty toxic. And it's like the same thing with any other type of cell. If a cell basically dies by necrosis, and then spews its insides outside of the cell, it's very toxic. Certain substances can't mix with each other, and that's where you have the macrophages that come in a nutshell, basically to come and like detoxify what's basically happening in there. It's the same thing for the microglia. And due to the release of their cytokines, because that's what happens when they phagocytose something, is that when they phagocytose, they're going to release cytokines, and when activated, that's what's going to cause inflammation. So, for example, like meningitis, that's probably something that comes up to my head. When you're dealing with meningitis or encephalitis, I think that would be a more proper term, yeah. So when you're dealing with encephalitis, think of encephalitis as the microglia having a huge role in the encephalitis part. With that being said, that's what the drawing looks like, and right below, I think that's the microglia cells in brown inside of a rat or a mice central nervous system. But anyways, moving on. Oligodendrocytes finally, and the neurolemocytes. Finally, the last type of supporting cells are these guys. The oligodendrocytes are mainly found in the central nervous system, whereas the neurolemocytes, or most likely known as Schwann cells, are found in the peripheral nervous system. So, to determine what you're looking at here, so you see the big blob right there, the big purple blob. Those are your oligodendrocytes versus the beads. They look like beads. Those are your neurolemocytes, which are... See, the oligodendrocyte is one cell that innervates multiple neurons at the same time, while the Schwann cells... I'm going to refer to them as Schwann cells. It's really easier than neurolemocytes. Neurolemocytes is more like the scientific term. But Schwann cells is because some guy named Schwann, I don't know his first name, but some guy named Schwann found this and decided, hey, I'm going to name them Schwann cells. Anyways, so the Schwann cells, as you can see, they look like beads, but each individual bead is a Schwann cell. Right? So you got multiple of them innervating an axon. So, in a sense, they both do the same thing, but they're found in different parts of the nervous system, whereas both of them provide myelinization of the neurons for rapid conduction and also reduce leakage. So, to explain what I just said, myelinization means that they will provide... they will wrap around the axon, and we're going to get to the axon, what it is shortly, but basically the axon is the tail, the elongated tail of the neuron. Basically, they're going to wrap around certain parts of the neuron, and again, provide, in order to provide rapid conduction and reduce leakage of the protein channels, right? Because you want rapid conduction, right? And the more rapid conduction that there is, the better transmission of information you do throughout the body. So if you have less of this myelinization, you get something called Gilenbare. Well, this is the one that's most likely well known. There are others, but a common type of disease is called Gilenbare Syndrome, which is the destruction of these of the myelinization of these cells. It's also an autoimmune disease because you're destroying these cells. It's basically your autoimmune system that just basically goes haywire, and it's like, hey, these guys are enemies, so we're gonna destroy them. That's basically what happens. We're not going to go into very much detail, but do know that that's one of the type of the diseases found via these cells. And then finally, I think I stated it before, but an oligodendrocyte can innervate multiple neurons at once, while multiple neurolemocytes or Schwann cells innervate only one single neuron. So basically, I wasn't going to go in there. I don't really want to go in there. But think of it as this. The oligodendrocyte is a bukkake. And the Schwann cells offer a harem. If you can put it that way. Best jokes of 2021. Meme Lord 2021. Anyways. Moving on to the main event, our attack damage carries the neurons, responsible for neurotransmissions as we know. And you pretty much find them anywhere in the nervous system. Again, making up only 10%, just remember that. Where you can find them around the central nervous system, the peripheral nervous system, the sympathetic autonomic nervous system, the parasympathetic autonomic nervous system, and the somatic nervous system. They're found pretty much everywhere. And I can't stress this enough. They're just unable to divide once they reach maturity. I don't remember. I have a blank, but I don't remember what the baby cell that can has 3 pathways that it can go through. It can either become a neuron, it can either become an oligodendrocyte or a microglia, or an astrocyte, I think. I don't remember what they're called, but anyway, that's the basic pathway that it takes. With that being said, the main takeaway of the neuron is basically that they are responsible for the synthesis and release of various substances such as adrenaline/noradrenaline, various releasing hormones. They all happen in the sorry, I have a blank right now for the word I'm looking for. I think it's hypophyse, but don't take my word for it. Anyways, the various releasing hormones, for example, thyrotropin releasing hormone. I hope that's the right one. TRH, basically. I hope I didn't screw that up. Basically gets released from the hypophyse, goes down to the adenohypophyse. And then release its in this case TRH stimulates the release of TSH that then goes to the respective endocrine organ, and then or the glial in this case to release that. But anyways, that's what happens. So various releasing hormones is one of the main jobs of the neurons. Then they can also release acetylcholine and others such as endorphins, but these are basically the main ones that I kind of thought of. There's many neurotransmitters that they release. But like I said, these are practically the main ones. And as you can see, this is what a drawing of a neuron basically looks like. It looks like a weird tree of life, if I can put it that way. It's like a weird alien with legs, if I can put it that way. Anyways, and as you can see, that's below in the cerebral cortex, what it looks like. Moving on. Before we can get to what the jaw, like, in depth, because what we're basically going to be talking about is going to be the release, how the neuron work and function to release these neurotransmitters in order to produce, like, movement in the body, for example, or receive and send information throughout the body. We're going to see that in a short while, but before that, we need to talk about the types of neuron that are found in the human body. Starting first with the one that you're seeing in the simplified drawing on the left, and on the right, we've got our multipolar neuron, which are mainly located in the central nervous system. And then moving on, we got the unipolar neuron. This guy doesn't have any dendrites, and we're going to get to what dendrites are in a second. But these guys, the unipolar neurons, are usually located in the cranial nerves and in the ganglia. Finally, we got the pseudo-unipolar neurons located in the peripheral autonomic nervous system and the sympathetic autonomic nervous system. And then finally, the bipolar neuron. These guys are mainly found, which are, as you can see, the bipolar neuron differ from the unipolar. I should have probably regrouped them together, but anyways, these guys, the bipolar guys, are mainly located in the eyes, internal ear, and nose. That's all where you're probably going to find them. And correct me if I'm wrong again. Moving on. This is where we get to our neuron physiology. And we're going to start first by describing every single part of neurons. As I stated before, our multipolar neuron is comprised of dendrites. Where are dendrites? You got that little, we probably call them hairs. Like crazy hairs. Or tentacles that spawn out of its, like, head, like a Medusa. Medusa has snakes, though. I should have probably said tentacles. That's so wrong. I'm so sorry about that. I've completely taken that out of context. But anyways, those are what dendrites are. They're like the snake hairs of the Medusa, right? Then we move on to the body of the cell, which is called the soma, or the cell body. I'm gonna call it soma. I feel like it's way more fancier to call it soma. Of course, at the center, you got the nucleus, the control center of what goes on in the neuron. That's your nucleus. Finally, moving on to the axon. The axon, actually, like I said, is its tail. It's that elongated tail, which is called an axon. It's the yellowish part. And then obviously you got the myelin sheath. That, like I said, from the supporting cells, they wrap around the axon part. They're kind of created. They are created by the oligodendrocytes in the central nervous system, and from our Schwann cells. In this case, it's Schwann cells that is being shown in the drawing of the image, that are found in the peripheral nervous system. And then in between, there's an in-between part, like the gap between from a Schwann cell to another Schwann cell, there's going to be a gap, right? And in between that gap, that's what we call the node of Ranvier. It's just a fancy word of saying, I don't know, man, these guys like to add their names to everything that they find, which is pretty understandable, you know? If I find something in science, I really want to have my name on it. But anyways, moving on to the, so basically you have the tail of the neuron, which is the axon, and then it divides itself into multiple tails, which are called synapses in this case. Those are called synapses, which is where the synapses, which is the spot where the neurotransmitters get released from. So with that being said, if we move on, now that we got the anatomy part out of the way, we can move on to the physiology of the neuron. So basically, a neuron's job is to trigger a neurotransmission. That's what we stated. But in order to do that, the neuron must be excited to do it, right? In order to commit something called an action potential, right? It's like basically trying to get an edgy teenager to go to the circus with you. You're like the 40-year-old daddy trying to take your kid who's like an edgy teen or like an emo kid, right? To get excited for the circus, right? Let's go to the circus. So that's basically what happens. It needs to be excited for it. There has to be a reason to do it, right? And to accomplish that action potential, the polarity of the neuron has to be superior to its excitation threshold. And we're going to get to that because there's a graph to understand before going into the action potential and everything. So, in order to satisfy the condition of the excitation threshold, there has to be a high amount of something called an excitatory postsynaptic potential, or EPSP for short, rather than an inhibitory postsynaptic potential, or IPSP for short. I didn't really mark it. That's a mistake in the presentation over there. But once the action potential reaches its synapses, the neuron will finally be able to release its neurotransmitters, and we're gonna see that right away. But before we get into the core of how an actual potential triggers and how it works, we have to understand this graph, and I'll try to explain it as easily as possible in my own words. I'll start first with saying how the action potential travels. So it travels from the dendrites all the way down to the synapses. And the way it does that basically is that, remember this. You have to remember this. You have to really remember this. I can't stress this enough. An action potential is always starts with a positive charge, and then is followed by a negative charge, because what happens is that you have to depolarize the neuron, and then you have to hyperpolarize it, and that hyperpolarization is the negative ions that are being transferred in and out of the cell. So basically what happens, if I can just like explain this graph really quickly, a neuron, if you take a voltage, if you like take some voltages and like strap it around the neuron to calculate the voltage, you're always gonna have minus 70 millivolts at rest for the neuron. With that being said, 70 millivolts for the neuron, that's where it starts at. That's where the graph starts at. It starts at minus 70 millivolts. And then, what you want to do is get that neuron so excited that it gets to that threshold. And that threshold, ladies and gentlemen, is the like trigger that says, holy shit, I have to neurotransmit. That's my cue. I'm excited to do something, right? And this is what happens. So by the intake of positive ions that get inside of the cell, that's what's gonna get that neuron to reach its excitation threshold. And the excitation threshold is usually about minus 50, minus 55, depending on the textbook that you have. The majority of them that I read took minus 55, so I'm going to take minus 55 as well. So once it gets at minus 55, oh boy, that neuron wants to send out as many neurotransmitters as possible, and that's what's going to happen. That action potential is going to travel in the axon, right? And every single time that action potential travels, it reaches plus 30 millivolts. And that tells the neuron I have to intake, actually no. The voltage gated ion channels open to let the intake of positive ions flow inside of the neuron, which is going to get the neuron all the way to plus 30. And then at plus 30, it's like, oh, okay, I'm reaching my limit. Let me shut everything down, right? I'm going to start shutting down the store. And that's basically what happens here. So if I can just explain this, the red line that you see, let me start with the red line. At minus 70, you got something called like the neuron which is at rest. That's its resting potential, which is always going to be minus 70. Then in blue, you got your EPSP. Remember what EPSP stands for? Excitatory postsynaptic potential, bringing the neuron up to the excitation threshold. And once it reaches that, that's where the green line comes in. The green line, it's our depolarization. Now we can talk about depolarization, where you got a lot of intake, a lot of positive ions coming in at every single part of the neuron's axon. And then once it reaches plus 30 millivolts, that's where the neuron says, okay, I've had enough. I've had enough of excitation. I got to close down shop. And then that's where the purple line comes in. The purple line is our hyperpolarization. And our hyperpolarization, I probably shouldn't because the yellow line actually has no has no significance. It should be in purple. I don't know why I put it in yellow. But anyways, the yellow line goes all the way down, hyperpolarization, and we're going to get to the reason why the neuron doesn't go back to minus 70. It goes back, actually it's going to go down to minus 90 millivolts, right? And that's called our hyperpolarization. Because what's going to happen is that at going towards plus 30, so if we go back to the green line, going to plus 30, the ion gated, voltage gated, that's the word I was looking for. Voltage gated ion protein channels are going to open up to intake. Like I said, it wants to intake as many positive ions. Once it reaches minus 30, those voltage gated ion channels will close, and then that's going to cause its hyperpolarization all the way to minus 90. The blue line then comes into play, and then from minus 90, it has to go back all the way to minus 70. And that's where our repolarization happens, because the neuron doesn't want to work harder to get back to minus 55. It doesn't want to work even harder to reach its excitation threshold. No, it wants to get as close to it as possible. So that's why it wants to go back to minus 70. So it wants to reach that balance, right? It wants to have that balance inside of its inside of it, right? That's what happens. And then finally, once it's at resting potential, that's where it goes back to minus 70. So with that being said, that's the ideal scenario that the neuron wants. And with that being said, let us now go in depth on how that actually happens. And with that being said, here I have a neuron, beautiful neuron, worthy of Pablo Picasso's signature. And here I'm going to explain what happens during the action potential. And the reason why I showed you the graph is because I'll also be drawing the graph at certain parts of the of the explanation, basically. So let's start things first at the top, right here, and at every single parts of the dendrites. So if we start at the top, let's assume, because the thing that happens is that you need other neurons, right, to already have scent or neurotransmitters. Let's say one of them is like here. Actually, that's a horrible drawing. Let me just redo this. Let's see that there's an axon right here. That's also a terrible drawing. Oh my god, what is going on with me? Let me try to do this. 1, 2, 3, 4. Like that. And then like that. Okay, that's a little bit better. Okay, let's assume that this synapse already sent. There's an action potential. It's sent its neuron and it's going its neurotransmitters, and it's going to this neuron here, right? Let's assume that's happening right now. Let's assume they already did it. So it's gonna send multiple. This is our neurotransmitters, and our neurotransmitters look kind of like this, right? Right? And what it wants to do, what these neurotransmitters want to do, is that these are ligands, right? These are like ligands, and what they're gonna do is that they're gonna go. Let's assume this is I don't know, adrenaline. Let's say this is adrenaline, right? Let's take the first one that we saw in the previous slides as adrenaline. So what adrenaline does is that it's going to go from here. It's going to go all the way to a ligand protein, to a ligand channel protein. It's going to go attach itself right here. And once it's in place, let's assume this guy attached over here. And once it's in place, once the ligand goes in place, it opens up the protein channel. The protein channels. And what happens is that, first of all, let's start with this guy over here in yellow. What happens is that this over here, it opens the gate. Sorry. So it opens the gate. Let's say that there's a gate over here. It opens its gate, allowing for the incoming of sodium. So there's a lot of sodium. As we know, there's a lot of high concentration of sodium outside of the cell. Outside of the extracellular space, there's a high concentration of sodium going on. And what that's going to happen is that there's a lot of sodium. If you open this, there's a lot of sodium that goes inside of the cell. And when that happens, if you got a lot of positive ions going into the cell, what does that do to the cell? It gets it more positive, right? And that's essentially what our our EPSP is. Excitatory postsynaptic potential. And that's what happens. Now, the other thing that we didn't talk about, that wasn't on the graph. That was the last slide I showed you was the ideal scenario for our neuron. But what happens is that there's also this ligand. There's also this ligand here that attached to this protein channel, this ligand gated protein channel. And then when that happens, there's two things that can either happen. These are negative ion ligand gated ion channels. And since they're negative gated ion channels, what happens is that once this attaches to there, is that inside, first of all, inside of your cell, you have potassium, right? There's a high concentration of potassium inside of the neuron. And what happens is that when this channel opens its gate, you see it just opened its gate, the potassium will leave the cell. And when positivity leaves the cell, what does that do to the cell? It makes it more negative, right? Another thing that happens, let's say we got another neurotransmitter right here. We got this guy right here. That just came and attached to that guy right there. Opened its gate as well. You also have chlorine, which is in high concentration outside of the cell. And since it wants to follow its gradient, its concentration gradient, it's going to want to go inside of the cell. It's going to go from the most concentrated, and the same thing goes for the sodium, for our potassium. Wherever there's a high concentration, it's going to want to go to the lowest concentration, right? Same thing for our chlorine. From the highest concentration to the lowest concentration. And what does that do if minus goes into the cell? It's going to render the cell more negative, right? And so we're dealing with a problem here. We're definitely dealing with a problem here, because if I go back to my graph and I do my graph again, let's say we got minus 70 millivolts here. And this is our excitation threshold, which is again minus 55 millivolts. What happens is that if I ever get, and I'm going to use different colors for this. I'm going to go with yellow because those are our positive ions. If I get an intake of sodium, which is positive ions inside of the cell, this happens. This right here happens. It's going to try to go up. But then I got either the exit of positive ions, and I also got the intake of negative ions. So what does that happen? This happens. You see that? So this here all the way to this here is an IPSP. And then this here to this here is an EPSP. And what you want is a more higher concentration of EPSP, because you don't want this to happen, right? You want this to happen in order for this to happen, right? That's what you want to happen, what you're trying to go for. And as such, in order to do that, you kind of understand the concept is that you want more EPSPs rather than IPSPs. And eventually what's going to happen is that due to the high intake of sodium inside of the cell, you're eventually going to have a higher concentration of sodium, and that positivity is going to cause the cell, the cell membrane, to become more positive. And that positivity is always going to travel. Let's say it entered here. Well, that positivity is going to travel all the way to what we call the trigger zone. So this is going to travel all the way to this zone right here. Actually, I'm going to highlight it. Actually, no. This is going to take a while to draw, but we're going to get to them really quickly. And another bad boy over here. And like another bad boy over here. And then let's say you got another myelin sheet over here with this nucleus. Like that. And so this is what's going to happen. So let's start here at the top of the axon. So there's an action potential going on. It travels here. You're at so if I redraw the graph, because the graph just happens to be, it's not just one graph for one neuron. It's a graph for like multiple parts that's happening outside of the neuron. So we got our little minus 55 over here, our minus 70. It just went here, right? That's what's going on. So there's like a lot of positivity going on, right? So what's going to happen is that, again, you have your high concentration of sodium. It's going to enter the cell because it wants that positivity to just go this way, right? And then at the same time, it also activates these guys, which is your voltage-gated ion channels, but for the potassium. And since you got a high concentration of potassium inside of the cell, it's going to want to leave the cell, making the cell more negative. And and if you remember what I said before, is that when the action potential travels, there's positivity that travels followed by negativity. And that's what's going to happen. So with that being said, this action potential, and we got our myelin sheet here, over here. We got this guy is the myelin sheath. Actually, there's an A, sheath. Courtesy of our Schwann cell. So nothing goes on here. Nothing leaks in or out of the cell. The action potential just passes through here. Passes directly to here. So we're gonna get to a little bit to the green popcorn a little bit later. What I just want to say here is that it's gonna travel again. The positivity is gonna travel again all the way over here. And then travel all the way over here. Same thing's gonna happen, baby. Sodium goes in. Sodium goes in. And then let's say sodium goes in here. Let's start from here. And then it reached like plus 30 millivolts, right? It's right there. When this happens, and as I've stated before, that's like the trigger that says, okay, I've had enough. I had enough of my dose. I'm going to start closing shop. And so what's going to happen is that the voltage-gated protein channels, protein ion channels, are going to start closing shop. So this is going to close, and it's going to do this. It's going to close its doors, meaning that there's no more incoming of sodium. And the same thing for the potassium. It's going to close its door like this, so that no more potassium can go out. The problem is and then it's slowly going to start doing this. The problem is that the slowpokes are the potassium voltage gated protein channels that take a lot of time to close down. And since they take a lot of time to close down, that's where you got your minus 90 millivolts. And now, unfortunately, you've got an irregularity in your neuron. And that's where our little green popcorn comes in. This little green popcorn right here is the sodium potassium pump. Because the irregulation that you have right now is that you have a lot of high concentration of sodium now inside of the cell, and you've got a high concentration of potassium outside of the cell, and you have to bring that balance back, right? You have to bring like that balance back. You can't just have that inside of your cell. And so that's what's going to happen. So the first things first is that the ions that are going to come out, it's going to pump out, this pump is going to pump out three sodiums in order to intake 2 potassiums, bringing back that balance. And then what's going to happen is that then you're going to have your repolarization. That's what I was talking about. So hopefully you can better now understand the graph. Hopefully it's way better. But moving on towards the action potential that travels again through the myelin sheet, and now let's say it's finally gotten to our 3 synapses. So that action potential travels all the way around the membrane. Travels all the way to its membrane. And then same thing for here. It just travels, all the way to the membrane. And then it reaches the synapses. What I like to call the synaptic button. And inside of the synaptic button is the same thing. Actually, what you're going to have is you're going to have these vesicles that are your neurotransmitters that contain your neurotransmitter. In this case, we said it was adrenaline. And so, in order to do that, in order to have the release of these vesicles filled with neurotransmitters, what you want to do is basically have that action potential travel, and then what's going to happen is that, see here, there's actually more protein channels. More protein, voltage-gated protein channels. And once the neuron, that action potential reaches this spot, and I made them in red because these guys are special. These guys are calcium voltage-gated ion channel. And you know what that means? Is that you also have, in the extracellular space, you also have a high concentration of calcium. And again, following its concentration gradient, or the inverse of it, a high concentration of substance always wants to go to a lower concentration of substance. So your calcium is going to want to intake and go inside of the cell. And that's what's going to happen. And the calcium is actually... I forgot to do a plus. There we go. And that's your calcium. what's going to happen is that this guy is going to head in between here, in between the vesicle, and the cell membrane. It's going to head in between here. And it's going to attract let's say the tentacles of the vesicle. The more specific or a more fancier name for that, it's called one of them. Actually, let me start over. The vesicles have tentacles coming out of them. Let's start from there. And those are called V snares. If I'm not mistaken, one of the V snares of the vesicle is called syntaxin or synaptobrevin. Whereas on the synapse button right here, you're also going to have like tentacles popping out of it, which are called T snares. And one of these T snares is called either syntaxin or synaptobrevin. I'm not sure which is which, but in any case, what's important to know is that the calcium is going to go in between these tentacles and is going to bind onto them, attracting them both towards each other so hard that they're going to fuse. It's going to make it as such that the cell membrane, that the vesicle fuses with the cell membrane. And then you have the beautiful, beautiful exocytosis inside of the extracellular space of these neurotransmitters. And that is how, ladies and gentlemen, you do a neurotransmission, and that is how neurotransmission works from a neuron all the way to another neuron. And these little neurotransmitters are also going to go do the same jobs. They're going to go to another neuron, and they're going to start the same concept. They're going to start the same action potential to that neuron. It's like a chain reaction, right? So that is the basic concept of an action potential, starting from the cell membrane over here, going all the way to the synapse or the synaptic button, releasing its neurotransmitter into the extracellular space, going to either another neuron, or in that case, you can either go to muscle, but we're not going to get into that. That's basically in a nutshell what happens. So with that being said, I just have a few shout outs to do, a few references to make. Thank you so much to the following references that have made me able to use these images. Some of the images come from Wikipedia, some of them come from this super cool website called Severe Medical Art. Thank you so much for you guys for having the Creative Commons attribution on your content. You have been really grateful, and I highly, highly thank you for letting me use your reference images in order to better make my viewers understand what I'm working with here. And then finally, for my information sources, there are two sources that I took my information from. First of all, was the Campbell biology book, which is super good and filled with super cool information about the neurotransmission of the neuron, how that occurs, and what happens inside of it. The second reference that I had taken from a book is Anatomy and Physiology book by the McKinley Books. Both of them were the French edition, because that's what I work with. And then finally, 3 separate videos. If you're really interested to learn more about how neurotransmission works, about the neurons, even in depth about the glial cells, I highly recommend watching a channel on YouTube called Ninja Nerd Science. This guy is super amazing. He's a physician assistant, a certified physician board physician assistant. Zach Murphy, PA. This guy is really amazing at explaining concept better in depth than I did. I just did a little introduction of the subject. He goes really in depth. So I highly recommend watching his videos about the glial cells, the ones that I mentioned in my videos in the stream. At the beginning of the slides. The second one was about the neuron anatomy and the function of the neurons, the one that I explained in the previous slides. And then finally, the rested membrane graded action potential. If you are curious and want to know more about the subject at hand, really, I can't stress this enough. He does really good content. With that being said, ladies and gentlemen, thank you for being part of the stream. Thank you for watching. You have been great. Viewers, if you've enjoyed the content, please hit the like, subscribe, and share button for more science information related videos. Thank you so much for watching, and I will see you beautiful people next time.